Hi, everyone. Welcome to the Lymphoma Research Foundation's update on relapsed refractory diffuse large B-cell lymphoma webinar. I'm Hope Avalone, and I'm the Senior Manager of Patient Education Programs here at the Lymphoma Research Foundation. During today's webinar, you'll hear from an expert speaker and you'll have an opportunity to ask questions. You can send those in at any time through the Q&A box at the bottom of your screen, and we'll address as many as we can at the end of the presentation. As a reminder, this webinar is being recorded. At the end of the program, I just wanted to remind you that you'll be prompted to complete a program evaluation, and we do ask that you complete this evaluation because we like to incorporate your feedback into future programming. But for now, let's get started. Thank you again to each of you for joining us on today's webinar. We'd like to first thank our sponsors of this webinar, AbbVie, Adaptive, Genentech, and Merck. And again, at the Lymphoma Research Foundation, our mission is to realize the promise of science to eradicate lymphoma and serve the community touched by this disease. All of our work is led by our Scientific Advisory Board, which is a group of 45 leading lymphoma experts from around the country. And so in addition to today's program, we have a variety of other programs and services available for you. The foundation has free comprehensive disease guides and fact sheets, which can be ordered or downloaded on our website, lymphoma.org. We also offer a variety of other in-person and virtual educational programming so that you can continue learning about the latest updates throughout the year. And as part of our digital resources, we have an award-winning mobile app called Focus on Lymphoma. The app provides helpful disease content as well as unique tools to help patients better manage their lymphoma. And this app is available free of charge in the Apple App Store and in Google Play as well. And in addition, the foundation offers a wide range of support services. So one example is our helpline, which serves to complement our patient education programs as a resource for both patients and care partners. The helpline has master's level trained staff who provide individualized information on all types of lymphomas, as well as information on treatment options, clinical trial navigation, and connections to resources such as financial assistance. And we don't have it listed here, but the number for the helpline is 800-500-9976, or if you feel more comfortable sending an email, that's helpline@lymphoma.org. And if you didn't get a chance to jot that down, you can just go to lymphoma.org and the information will be there for you. So in addition to our helpline, we also offer a peer support program called the Lymphoma Support Network. And through the Lymphoma Support Network, we can connect patients and care partners with others who have been through similar experiences for that emotional support component. And lastly, I just wanted to briefly highlight our upcoming West Coast Lymphoma Workshop in Los Angeles. Our lymphoma workshops are one-day educational programs that provide the latest information about lymphoma, current treatment options, as well as patient support issues. And so this workshop will include a specific breakout session on diffuse large B-cell lymphoma, and it will be held on Saturday, September 14th. So you can view the agenda and register for that program at lymphoma.org/West Coast Workshop. And we have a wonderful program planned for you today, and I'm so honored to introduce our speaker, Dr. Tatiana Feldman. Dr. Feldman is a hematologist and oncologist at Hackensack University Medical Center, where she is the director of the T-cell program. Thank you so much, Dr. Feldman, for speaking on our program today. I will now stop sharing my screen and turn it over to you. Looks great.
Good afternoon, everyone. Hope you can see my screen, right?
Yes, looks perfect.
Okay, thank you, Hope and LRF, for inviting me for this program, to present this program. So my goal is just to go over about diffuse large B-cell lymphoma and what's going on in the field, which, as a matter of fact, is quite exciting. Before we start, please, if I'm saying something which is not understandable because the doctors are guilty of using the technical language, sometimes forgetting that it's not really used by the patients and the family. And I can see that sometimes at the end of discussion, you realize you have to explain things in more lay terms. So I'm giving the list in the beginning. If you hear me saying that, so you're following, and I'm sure you probably already know those terms. What we use as a to define our therapies numbers, at least, as lymphomas frequently need to be treated once and sometimes more than once. So when we say frontline therapy, it means the very first time the lymphoma is treated. Relapse, meaning lymphoma came back after therapy. Remission means that there is no evidence of lymphoma. Remission becomes cure when it lasts for a long time, usually for diffuse large B-cell lymphoma. If remission is lasting for 2 years, then it's probably cure. It's highly unlikely to relapse afterwards. Salvage therapy denotes basically anything which is given to put lymphoma back in remission, but not the first time. And then second line, third line, that's exactly what it means. The second time lymphoma is being treated with something different than initial therapy. And a bridging therapy is some therapy which we give just to control the lymphoma with an idea that we'll use something definitive, either transplant or CAR therapy, but we need just to shrink what we call tumor burden to make less of a disease present. Then there is also a term called lymphodepleting therapy. This is something new. That's what's utilized not to treat lymphoma, but it's a chemotherapy, which is given right before CAR T-cells. We'll talk about this later. And a conditioning therapy, it's usually the term using before the stem cell transplant is being used. So what is lymphoma? Lymphoma is a blood cancer. All cancers are classified based on what is the normal cell which made a mistake and gave rise to the cancer. So the normal counterpart, or in other words, cell of origin, COO, is a lymphocyte. And lymphocyte is a white cell which lives in the lymph nodes and a part of our immune system, which is really very complex organ, tasks with the protection of us from hostile environment, whether it's viruses, bacteria, parasites, and cancers. Cancers which unfortunately start with everyone. We all have cancer cells, actually. They get killed by our immune system, and lymphocytes actually play a very important role in that function. So, and there is an exhaustive classification of lymphoma. This is some simple kind of a graph showing the subtypes. So lymphomas are split into two major classes. One is Hodgkin lymphoma and then non-Hodgkin lymphoma. And there are about 70 plus different subtypes of non-Hodgkin lymphomas, and diffuse large B-cell lymphoma is part of it. So you can see it on the right? So diffuse large B-cell lymphoma, the word B means it starts from B lymphocytes, because also we have T lymphocytes, which give rise to T-cell lymphomas, completely different ball games. Majority of lymphomas start from B cells, and diffuse large B-cell lymphoma is actually one of most common lymphomas. So it's one of the most common out of all lymphomas. And lymphomas generally constitute about 4% of all cancers in the United States. It is disease of elderly people, just like any cancer. Its median age of diagnosis around mid-60s. However, it can affect young adults as well. The incidence is about 35,000 new cases per year. And this is an aggregate number for everyone, all stages for the last 5 years. And from Sears database is about 65%, which is actually quite a good number. Why do we get lymphomas? I get those questions, every patient asks, why did I get lymphoma? I didn't smoke, I didn't drink, why did it happen? So most likely age. As we all get older, the cells get older, make mistakes, and immune system is particularly prone to genetic mistakes, because the way how it functions, it needs to mutate itself to mature the lymphocyte, so it becomes very, very highly specialized cells, which will only kill a particular offender and will not make mistake of killing everything around it. Then it makes mistake with age, and mutations accumulate with age and develop into lymphoma. A second major risk is actually what we call immunodeficiency. So when immune system is not fully competent, and one of the example is solid organ transplant. Patients are on medication to calm it down, so the rejection will not happen. And it's a well-known phenomenon with development of particular lymphomas, HIV, autoimmune diseases where some immunosuppressive medications are utilized. And then environmental exposures. There's some more knowledge about herbicide, pesticide response slightly increasing the risk. It's not exactly hereditary disease, meaning that we don't have mutation like a breast cancer, which if this mutation is present and confers very high risk of lymphoma, but the incidence is slightly higher in a first-degree relatives. It's still maybe 3 times higher. However, the absolute incidence is so low, then I wouldn't worry about high risk in children or siblings. And I would like to make a comment about environmental exposures, that the official funds created, for example, for veterans exposed to Agent Orange, and also September 11 fund for people who worked at the sites of September 11 explosion, that they actually, if those people develop lymphoma, there's a particular compensation available. So how do we make a diagnosis of lymphoma? And it's very important to have an expert pathologist. So the simple one, H&E stain, you can see this blue colored large box, which has all large cells kind of looking all the same. That's initial, and pathologist just looks at this picture and making an opinion what it could be. And that's how it was 50 years ago, 40 years ago. However, as science becoming sophisticated, there's many more techniques which we utilize. The next one was immunohistochemical stains, which basically like a fingerprinting of lymphoma cell. You can see brown, it's a brown color and a blue background. So brown is positive, blue is negative. So, and it said CD20. Keep this number in your mind. We'll talk about this later. So all those cells which you see here in blue, now they're all having this marker called CD20 on their surface. And basically all those markers on the surface is a means of lymphoma cells and the normal lymphocytes of communicating with an environment, getting signals, talking to others, and basically behaving based on an information they receive. And in the last many years, we have molecular studies such as genetic studies looking into mutational analysis, even at much basic level into lymphoma, see what are the genetic mutations present, and the test which do it, it's FISH or next-gen sequencing. Why is it so important? So the pathologist not just need to establish correct diagnosis, which is critical as therapy depends on that, but also it needs to help oncologists to identify the best therapy, because diffuse large B-cell lymphoma is actually a spectrum of the diseases. And this is just a list of a classification, WHO classification, that's a Bible of oncologists, which classifies all malignancies, including lymphoma. So DLBCL, it has multiple different subtypes just based how it looks. They also have different behavior and different responses to therapy. And in the last 20 years or so, the molecular subtypes also developed. So Hans classification divides DLBCL into germinal center versus non-germinal center, or ABC. Why is it important? Because the prognosis is slightly different between both. And then certain novel therapies, they work better for non-germinal center and don't give any benefit to germinal center. So it's important for the oncologist to know that when you're deciding how to treat. And this is not present, but it's rapidly developing, and I hope it's a future is a molecular classification based on next-gen sequencing, where you basically create a picture of genetic mutations. And this is, I put this unreadable graphs of kind of what we are going to see in the future when there's a biological classification and we test thousands of genes and artificial intelligence analyzes it via machine learning and basically spits out a particular picture of that particular lymphoma. And with time, we will know which drug works better, etc. So we can cure everyone. That's basically the goal. So, and with that, I would like to move to the part of talk, how do we treat the lymphoma? So diffuse large B-cell lymphoma is kind of controversial disease. On one side, it's life-threatening. It's type of a disease, it's all or nothing. You cannot live with diffuse large B-cell lymphoma. It's not going to sit and wait. We need to kill it. But it is highly curable. Some lymphomas like CLL, we don't need to treat and people never have to deal with that in their life because it just doesn't grow. Diffuse large B-cell lymphoma grows. So it's highly curable disease. And what is the chance of cure depends on several parameters. Some of it is lymphoma, stage of lymphoma, molecular pathology, some lab value at the time of a diagnosis. Some of it is patient-specific, such as age, comorbidity, what we call performance status, meaning how functional and strong is the patient, which tells us how well the patient can tolerate the treatment. And based on all these factors, including patient goals, the oncologists decide how to treat. And obviously, the goal of therapy is cure. We have to cure the patient. Sometimes we cannot cure, but we still can give very meaningful extension of life with a good quality of life. And I would say that quality of life is very important. So the therapy goal is to kill lymphoma and a patient can continue with a normal life. And I think what I'm seeing in the last decade, even if lymphoma comes back and we're not able to cure it on a first attempt, we still can cure it on a second attempt. In old days, it was very difficult to cure this lymphoma on a second attempt, on a third attempt. But with all novel therapies, we kind of keep pushing the bar. And I have many patients who doing just fine. And lymphoma may be coming back, but a new drug is developed, which is making a big difference. And I would like to show this graph, which I created myself. I'm not an artist, so it kind of looks not exactly smooth, as you could see some other places, but at least it gives you an idea. Actually, the very first immunotherapy was in 1975 when allogeneic stem cell transplant developed. So it's a bone marrow transplant from someone else for the patient. And then we had a very long period of over 30 years with chemotherapy, which is curative, but that was the only options. And the problem with chemotherapy, it has a high cure rate when you do it first time. However, when diffuse large B-cell lymphoma comes second time, the cure rate is much lower. And third time, the chance of cure with chemotherapy alone is close to nothing. And a major breakthrough happened in 2006 when antibody against CD20, to mention, you saw the picture, all lymphomas, most of B-cell lymphomas, including DLBCL, they have this CD20 protein, which is like an anchor on the surface of the cell. So rituximab is not chemotherapy at all. It's an antibody. We all make antibody, which are proteins designed to attach to very particular target and a function as a part of the immune system when it tries to kill some invader. So it's artificial antibodies, which attaches itself to CD20 and it provokes immune system to kill it. When that got invented, and in 2006, it was approved as part of chemotherapy CHOP, which was standard of care, it immediately increased the cure rate by about 15-20% in frontline setting. So that's, I think, was a seminal discovery. And then 10 years later or so, another, I think, major discovery which happened is the approval of CAR cells. CAR means chimeric antigen receptor T-cell, and 19 means it's a CD19. That's this other anchor on the cells which this CAR cell reacts to. And CAR is basically normal T-cell, which is a little bit manipulated. We'll talk about this later. There are 3 approved products: axi-cell, Bispecific, and liso-cel in diffuse large B-cell lymphoma and some other lymphomas. Pembrolizumab is also immunological intervention, basically makes immune system react again and kill the cancer. It was approved for particular type of lymphoma, primary mediastinal B-cell lymphoma. Then couple of novel method of administration of chemotherapy, such as antibody-drug conjugates, got approved, polatuzumab and loncastuximab. They're very good drugs. I'm not as excited about them as about CAR and bispecific T-cell engager because I don't think they're curative. But nevertheless, they can provide a very meaningful responses. And finally, the latest addition in 2023 is bispecific T-cell engager. I will talk about this later, but it's another antibody, which is dual arm antibody, which binds lymphoma on one side and T-cell lymphoma on the other side. It's a little bit too soon to tell, but that could be a potential curative intervention, just like CAR cells. And on the bottom, there is data from Sears, which shows on the top, it's an incidence of lymphoma from 1992 to 2022, which is about the same, maybe a little bit more. But what's interesting that you can see this mortality curve, which was solidly at the same level at about what the mortality rate per 100,000 sitting at 3. And then it started to shift down. And about 2000 and maybe a little bit later, it solidly started to go down. And I think that we're seeing finally the results of a scientific conquest bearing fruit of really changing the life of our diffuse large B-cell lymphoma patient. So this is what's kind of an algorithm when the doctor decides how to treat it. So this is what we do for diffuse large B-cell lymphoma when it comes back. So and there's some numbers that I would ask you to ignore. It came from a blood journal, and those percentages are somewhat confusing because they're not talking about the chance of cure. They're talking basically about the number of patients. So in old days, if lymphoma comes back after first therapy, if patient is in a good shape and young enough, the standard of care was to give some kind of chemotherapy to achieve response and then solidify it with autologous stem cell transplant. Autologous stem cell transplant, basically, it's a high-dose chemotherapy which wipes out lymphoma, and the stem cells or bone marrow is preserved in advance, so it's not exposed to this high-dose chemotherapy. It sits in a refrigerator, and then the cells are infused back, so they repopulate the bone marrow. And that increases the chance of cure, as the chance of cure from chemotherapy only on second attempt is very low. However, with rituximab invent, what we realized that if lymphoma comes back within 12 months of initial therapy with rituximab-containing regimens, then this option with a transplant is not really that great anymore because chance of cure was sitting around 20%, still the case. And that's where that's where CAR T-cell therapy came into play. This is immunotherapy. They don't care about chemosensitivity. Our T-cell can kill everything. And this is a new standard of care. So patients who relapse within 12 months after initial therapy should be evaluated for CAR T-cell. And majority of the patients, so 70% is probably more, are eligible. When I use the word eligible, meaning they're good to get it. And in case of stem cell transplant, it's a rough therapy. Most of people of old age or there's a lot of comorbidities, they wouldn't tolerate it, autologous stem cell transplant. So it was one reason patient couldn't get a stem cell transplant. The second reason, if lymphoma is not in remission, the stem cell transplant wouldn't work well. So that's another reason not to be eligible for stem cell transplant. However, this eligibility is kind of different. It's so much better tolerated that pretty much everyone can get CAR cells. And also patient does not need to be in remission for the CAR cells to work. They kill everything, as I mentioned. So, and that's potential curative option. And then if CAR cells don't work, or we need to shrink lymphoma before giving CAR cells, then there's a slew of other options which we can now utilize with the whole idea that we can cure from clinical trials, more immunotherapy, drug antibody conjugates, either polatuzumab or loncastuximab, some small molecules, selinexor, lenalidomide, combination of tafasitamide and lenalidomide. And what's a note on this slide, because this publication from 2022 is bispecific T-cell engagers, which really just got approved last year. And ultimately, if patient is not in remission after BiTEs or bispecific T-cell or CAR-T, young fit patients who were able to put into remission after several lines of therapy should be evaluated for allogeneic stem cell transplant, which is kind of ultimate immunological intervention. So that's basically another slide kind of to repeat myself. There's a slew of options which we can use. And again, it may not be curative, but it gives this time, if anything, of putting lymphoma in remission. And a modern age, if someone is in remission for a year, and there is a new drug comes in which can change the patient life, and we see more and more of those patients. So I think the disease will make a major difference. So with this, I would like to move on to talk about these novel therapies which we're utilizing. So this, I would call it the age of immunotherapy. So what are CAR T cells? It's the first CAR T cell was approved already about 7 years ago, as a matter of fact. So T cell is like a policeman. It's supposed to participate in killing cancer cells. That's what we care about for our purpose. And it's a very complex process because we do not want those T cells killing indiscriminately. So it's a big dance which involves a lot of immune cells and important cells called dendritic cells. That's actually a big cell which comes and chews up the cancer cell and then puts bits and pieces of this on its surface, and a T cell sees that, and it kind of dendritic cell giving instruction to a T lymphocyte, all right, this is a bad guy, go kill it. And there are many checkpoints and balances, so there are no mistakes made, and there are many signals which needs to be fulfilled in order for T cell actually go and kill the cancer cell. And the problem is the cancers are very good at interfering with those signals, pretending they're not cancers, and creating substances which makes T cell kind of impotent. So that's where chimeric antigen receptor comes in. It's an artificial construct, so it's a genetic construct which is inserted into normal T cell, which derive from the patient, so autologous CAR T cells from the patient. And basically, it puts all those signals, all those several signals which needs to be present for T cells to get active, they're kind of all in one. So it's a self-stimulating active cell, which has a very particular goal, only to kill cells which has this marker anchor on its surface called CD19. And that's our lymphoma cells, as well as some normal lymphocytes. And that's a nice cartoon kind of showing what happens. Your CAR T cells, when it's infused into the vessel in the blood, it starts proliferating, gets out of the vessels, and starts seeking out malignant cells, it keeps proliferating and keeps killing it. And a major difference from chemotherapy, for example, it's a living therapy. Those cells are alive. And in the clinical trials, they were detected actually up to a year from infusion. So they're there doing immunological surveillance. And if malignant cells start reappearing, it kills it again. So that's a major advantage. So the several products, Yescarta, Breyanzi, or Kymriah, or Axicell, Lysacell, and Tisacell, they all approved in a treatment of diffuse large B-cell lymphoma in second and third line. It cures about 35% of patients who really had no chance before, multiply relapsed, and patients who get it as a second line, the cure rate is probably even higher. And what we noticing that patients who in complete remission as per PET scan, so no evidence of lymphoma on a PET scan, at 6 months, most likely are cured because we don't see relapses. It's fairly well tolerated, and there are some downsides, however. So it's not a drug you can take from the shelf. You need to create it. And unfortunately, the creation process involves administrative steps, such as getting insurance approval. Insurance needs to make agreement with the hospital, and depending on institutional insurances, it can take up to 2 weeks. And diffuse large B-cell lymphoma cannot wait. And then you need to collect the cells from the vein. So this little diagram here. And then those cells, when we get approvals, then those cells go to the factory when they get manipulated and created. Many millions of those cells, they need to be grown. This process takes, depending on a company, anywhere between 14 to 22 days. And when cells are created, they're frozen and they sent back to the cancer center. And the cancer center will administer them. It usually will involve administering of chemotherapy. It's called lymphodepleting chemotherapy, kind of to create the space so those cells don't disappear when they're infused, because even though they're self, they're still a little bit different, and the immune system can just reject them. So this chemotherapy is given, and then those cells are infused. So this whole process can take between 4 to 6 weeks. And patients with particularly aggressive lymphomas, they really cannot wait that long. It's not a therapy which can be given in small doctor's office, and you need a major institution to administer. So people who live in big states with a lot of big institutions, they in a way have advantage. But I think United States has a lot of rural areas with not an easy access to major institutions. So that's one of the problems. It does have a couple of interesting toxicities, which are very manageable and transient, but they're scary and very unusual, and it's scary to the doctors as well. It's called CRS and ICANS. We'll talk about this. And also, the person needs a caretaker for the first 30 days. So people who are single, and we'll be surprised how many people actually don't have caretakers. It could be a challenge. So why do CAR therapy fail? Cancers are very smart. They start hiding CD19. Then sometimes those T cells, they're beaten up by prior chemotherapy, so they're not a good quality. And sometimes lymphoma is just too bad, and it grows too fast. So we're learning more and more about failures, and there are more and more research happening trying to improve on those CAR cells. So now they're two targeted T cells. We have a couple of targets, because obviously lymphoma cell expresses many targets, CD19, 20, 22. We can improve T cell quality by taking those cells from someone else. So it's allogeneic CAR cell. So it's made in advance. So you don't have to have this waiting time because those cells are ready. They're natural killer cells. It's another type of cell which can kill cancers. There are some tonics for T cells, some medications which could be given together with CAR cells. We can optimize production, et cetera, et cetera. So it's a very active field, a lot going on, and we're going to see results hopefully very soon. A couple of words about CRS and ICANS, because a lot of time, particularly when CARs got introduced, patients and community doctors who are sending their patients to academic institutions for CAR cell, they were kind of afraid of them because when they saw those CRS, so CRS or cytokine release syndrome, it's basically like an infection. You have a million of superactive T cells coming in, and they all start killing lymphoma. They must introduce, it's not collateral damage. That's how they work. Fever is very common, actually. So that's CRS, but it goes away. More severe CRS, we call it higher grade CRS. like sepsis picture, when low blood pressure develop, low oxygen level, leading to organ failure, just like in sepsis, it's seen, but it's not that frequent. And nowadays we know exactly how to deal with this and their medication. So it's really not a major issue, and it's even CRS happens, it's reversible. But that's the reason why some CAR cells needs to be administered in a hospital for the observation. And then ICANS, it's immune-mediated neuroencephalopathies. Basically, it's a very strange phenomenon. Probably those cells go into the brain because lymphoma cells, I'm sure, is there. And they start causing some symptoms. No one understands this phenomenon actually that well, but it could be very mild, and most of the time it's actually very mild, such as headaches and some sleepiness. But if it becomes severe, people may lose ability to speak or ability to write or develop confusion. And the worst case scenario is a brain swelling and seizure. So, and that was pretty scary. However, again, the severe toxicity is very low. And it happens at a very predictable time, usually 5 days after the infusion. And nearly 100% of the cases it resolves. But it could be very scary, particularly, we had patients and it happens very suddenly. It can deteriorate very suddenly and improve very suddenly. But suddenly patient is no longer talking and completely unaware of environment. But again, these are very well known. We know how to manage it. And this type of toxicity really resolves. And the beauty of the CAR cells that really, whether patient is young or patient is elderly, they really tolerate it very well. And there's tons of data available showing that elderly patients who get CAR cells do just fine. So I think that also contributed major improvement into elderly DLBCL, which I think in an era of chemotherapy only was shortchanged, because a lot of people of old age, they would not tolerate chemotherapy, but they are tolerating this. There are some other toxicities which doctors are aware. They really don't interfere much with a lifestyle, and it tends to resolve. With that, I would like to talk to the next exciting thing, bispecific T-cell engager. So what is that? It's basically an antibody, just like rituximab is an antibody, but it has two arms. Let me see my mouse. Okay, so one arm will bind anchor, which is present on a tumor cell. In case of lymphoma, is a CD19, CD20. The ones which are approved bind CD20. And another arm which binds CD3. CD3 is an anchor which is present on a normal T cell. And it sounds simplistic, but cannot beat the clinical data. The drugs work. It looks like it just brings T cell, normal T cell, in a vicinity with malignant cell, and it creates immunological reaction when it pulls some other immunological cells, and the killing process starts, and a T cell will kill the tumor cell with the help of this antibody. So it kind of redirects naive T cell to eliminate malignant B cells and engages the immune system. There are several antibodies which are already approved. Glofitamab, mosunetuzumab, and epcoritamab are approved, and odronextamab is still at the research phase. And there are antibodies with a CD19 as a target. All these antibodies are CD20 as a target. There's still clinical trials ongoing. And blinatumomab is an old antibody which is utilized in ALL, not diffuse large B-cell lymphoma. So just a quick slide to show you that how active those agents. So you can imagine that those drugs were tried in patients who had a lot of chemotherapy. They had everything which is available to medicine, including CAR cells. You see almost 40%, anywhere between 30–40% of patients had CAR cells and relapsed, unfortunately. And this shows epcoritamab, glofitamab, they're already approved for diffuse large B-cell lymphoma. This is trial still ongoing. And you can see that overall responses are quite high, with 60% with complete response rate is around 40%, which is quite actually remarkable. And mosunetuzumab is the only one which did not have a very high activity in diffuse large B-cell lymphoma. However, it's the easiest drug to give, and it has excellent activity in follicular lymphoma, and that's why it's approved in this indication. And what we're seeing, although the data is very young, that patients who achieve complete responses, they appear to be in complete responses. And some of those patients move on to allogeneic stem cell transplant just because the doctor doesn't know how long the response will last. But what we see that patients who achieve responses, and now we're utilizing molecular analysis of residual disease, that they don't relapse. So I'm very hopeful that this will be a next curative option. And the bispecific T-cell engagers are less toxic than CAR-T. So they also do have this CRS and ICANS phenomena, but the incidence of ICANS is pretty much negligible. And beyond the headaches, we rarely see anything else. We don't see any lethargy or confusion, etc., just headaches. And CRS also, mainly we see fevers. We don't see more than that. One antibody is administered subcutaneously, epcoritamab, so some patients get injection site reaction, anemia, neutropenia, infections, kind of a usual panel, but generally speaking, they're really well tolerated. They're not chemotherapy. They're what I would call good quality of life drugs in a way that people feel fine with them. Disadvantage as opposed to CAR-T. CAR-T is a one-time deal. You get it, and this is it. If it worked, you don't need to get any more therapy. Doesn't mean that you don't need to deal with the doctors, but at least you don't need to get any therapy. With the bispecific T-cell engagers, it's a therapy. It's off the shelf, so you don't need to wait to get it. But you have to come at prescribed intervals to get it administered, just like as any other drugs. The only good thing that the therapy most likely is going to be definitive for a particular period of time. And just kind of the first three columns are the bispecific T-cell engagers, and these are CAR-T cells. And as we can see, it's not comparable in a statistical way, but kind of a very similar result. So it's we're getting more and more options. And the toxicity, probably bispecific T-cell engagers are less toxic than CAR-T and much easier to administer. They could be given in a community oncology settings. So we just need to wait a little bit more to see whether the bispecific T-cell engager actually have the cure rate, just like CAR-T cells. So I would still consider CAR-T as my first option and a bispecific as next, rather other way around. But we have options, and we already have knowledge that bispecific T-cell engagers do work after using CAR-T and CAR-T failure. And CAR-T work after failures of bispecific T-cell engager. There were some clinical trials showing it. So I probably would spend a couple of more minutes to talk about what else we have available. So antibody-drug conjugates. These are basically different way of administering chemotherapy. Chemotherapy has a narrow window between becoming too toxic. So it's a window between it being active and being too toxic. And what drug antibody conjugate does, it allows us to give much higher dose of chemotherapy, because it's basically a delivery of the drug, which is attached to the antibody. An antibody binds particular anchor on a lymphoma cell. So it gets inside the cell and kills the cell when the drug is released. And while the drug is in a vein, it's inactive. So some examples in lymphoma: polatuzumab, lancastuximab, and rituximab vedotin. And the data, they basically achieve response in about 50% of a patient, give or take, with complete responses, and up to 35%. And there is a meaningful duration of responses, anywhere between 3 months to over a year. And because they're less toxic, patients can receive long-term treatment. I don't consider this type of therapy curative, just like CAR-Ts or BiTEs, but nevertheless, it's the very good addition because it's not a regular chemotherapy. They're well tolerated. They also play a big role in a bridging therapy. This 4 to 6 weeks when patient has to wait for the CAR cell, you can give those drugs. They have low toxicity to trying to control lymphoma growth. Another very interesting combination is naked antibody against lymphoma, a CD19 antigen, tafasitamab, plus lenalidomide. It's an oral agent with a fascinating activity. And overall responses were 60%, with complete responses of 40%. It's not as active in patients who had more than 3 lines of therapy, but elderly patients who, for some reason, cannot go on CAR cell or bispecific T-cell engager, that could be a good option. I had patients who would get this regimen for a long time because it's really not chemotherapy very well tolerated. There's a recent publication. We kind of have to see a little bit more data, but it's a combination of lenalidomide plus rituximab, which is CD20 antibody, plus drug antibody conjugate brentuximab, which technically speaking does not bind to a target on B cell necessarily, but I think it's really immunological intervention. So it looks like overall responses 60% with complete responses 40%. I cannot really say anything about how long it works because trial is too young. So I think to summarize, we do have a lot of activity, and I think we're really meaningfully changing our patients' cure chances in a relapse settings. With the CAR-19 are still the best potentially curative options. With bispecific T-cell engagers, a new kid on the block, we need to wait a little bit longer what the trials show, whether it's a curative options. And we do have a lot of novel therapies, which maybe they're not curative, but they definitely help to keep responses and maybe to wait for something new to come up and save the day. And of course, there's a very promising landscape of clinical trials. Again, a little bit of a reminder, clinical trials are classified into phase 1, 2, and 3. Phase 1, it's an early trial with main objective is to see how toxic is the drug and what the dose should be. However, modern phase 1 trials are different from old ones, which would pile up any disease, whether it's breast cancer, lymphoma. But modern phase 1 trials, they really already, it's molecules which are designed based on disease state and based on molecular findings and genetics. So we already have an idea that this drug should work for lymphoma. So already phase 1 is becoming much more specifics. We're not running those big trials where every patient goes on this phase 1. You have no idea whether it's going to work or not. We have a lot of phase 1 trials which are very meaningful. And then phase 2 is to determine efficacy, to see whether this drug is really working at the dose level, which it's safe. And the phase 3 is rarely done in oncology because it is difficult to find standard of care. But it's a comparison, head-to-head comparison of a standard of care with investigational agent. And I do not know if you see it well. I'm sure you guys all know it. But there is clinicaltrials.gov and cancer.gov, that major website maintained by NCI, which lists every clinical trial. Investigator is obliged to list a clinical trial with this website. And there is a lot of information which will tell you what is a trial, which disease, what's the line of therapy, and it'll give a list of institutions which run this particular trial, and it will give a phone number of coordinator if you need to reach out. So, I will need another hour just to list clinical trials in diffuse large B-cell lymphoma, but kind of the what's going on, we're trying to move CARs and BiTEs into frontline settings to see if we can cure everyone, so we don't need to do it second or third time. And these are very active regimen. So that's what usually happens in oncology. It starts with relapsed patient, and if it's something very active, we start running trials in frontline settings. And the results are very promising. We need more time. Then finding novel CARs and novel BiTEs, right, to improve those T cells. There are a lot of trials going on allogeneic CAR cell therapy, natural killer cells, autologous CAR T-cell with different targets, et cetera, et cetera. There's still a lot of novel molecules as we understand lymphoma workings better. There's new molecules called degraders, which basically eliminate particular proteins, and you can design it to eliminate the protein which lymphoma is dependent on. So you eliminate this protein, a lymphoma cell dies. Cerebron inhibitors, and list goes on. And also, because novel drugs are no longer chemotherapeutic drugs, their toxicity is much less. We need to identify active combinations, because we can put several drugs together and create this magic potion which will cure lymphoma. So to summarize, I think it's my last slide, is what I see, as I've been practicing only lymphoma for many, many years already. I see that clearly many more diffuse large B-cell lymphoma patients are cured. With all the scientific progress is really playing out and producing fruits, explosion of immunotherapy agents. And another very nice thing, you know, this elderly patient is disappearing. We always say that it's not an age, but how strong is the person. But in chemotherapy era and stem cell transplant era, age definitely was precluding treatment for some people because they would not be able to tolerate toxic regimens. All those novel regimens, CARs, BiTEs, etc., elderly people tolerate them just as well as young adults, and it's bringing a meaningful cures and changes. So with that, I will stop and will be glad to answer questions.
Perfect.
And that's an institution which I work for, John Tura Cancer Center. And the designer are much better than I am when I put those beautiful.
It is
words.
beautiful. Thank you so much, Dr. Feldman, for walking us through your presentation. We're going to get to just a few questions here before we wrap up today. We already have some questions in the Q&A box, and I'm going to read some off to you. So the first question I wanted to address, this participant notes that you address some hereditary factors, and they say, myself and my husband and my mother all have had DLBCL. They currently have recurring DLBCL. And they're wondering, would you still not be too concerned about their kids?
Oh, it's an excellent question. Husband and wife, unless you are first-degree relatives, that's not hereditary. But I agree the concern over the children when both parents and a grandparent had it. The question, if you have way too many family members, I would look into the tree, whether there are other cancers in the family, colon cancer, sarcomas, brain cancers, because there are some hereditary syndromes like Lynch syndrome, Li-Fraumeni syndrome, which generally predisposes to cancers. Usually they're medical geneticists which address this question. So if there are many family members with other cancer in the first degree particularly, I would look into it. But we still don't have a strong hereditary connection. So as I mentioned, probably the incidence of lymphoma will be 3 times more in a first degree relatives rather than general population. However, if you look at the number gains, right, the absolute incidence of lymphoma is so super low that the chance of developing lymphoma is still very, very low. That's number one. Number two, to make you feel better, maybe, breast cancer, colon cancer, if you don't detect it early on, there is really no cure in a metastatic settings when it metastasizes. So the best option is surgery. You need to cure. Lymphoma is different disease. If someone has lymphoma and it's not causing any symptoms, who cares? There is no difference when you treat lymphoma. If you treat it a little bit later when you noticed it versus a little bit early, it doesn't matter for most of lymphomas. DLBCL, obviously, you not want to get too sick from that, but most of people will notice something is wrong early enough to intervene. So we're not in this situation that, oh, I missed it. I should have detected it earlier. So that's not the situation with lymphomas, because surgery is never a treatment option. It's for diagnosis. So that's why I wouldn't worry, because what are the options? You're not going to do CAT scans on a regular basis out of fear. You accumulate radiation. So the only thing, so I wouldn't be concerned. It's not like hereditary breast cancer with a 70% chance of having breast cancer with a BRCA mutation. Um, so I hope that answered the question.
Yeah, I think that's reassuring. Thank you. Another question that I want to address here, what is the relapse rate of triple hit lymphoma, and are there unique treatments for triple hit if you relapse after chemo?
So I think triple hit lymphoma, I'm sure, just it's a for people who may not know what it means, it's a particularly aggressive lymphoma which has several mutations. One is it has to be MYC, translocation, not mutation, translocation with either BCL2 or BCL6, basically creates the animal which grows very fast and chemo resistant. And usually CHOP, Rituxan CHOP, does not have much of a role because cure rate is very low. And the problem with this lymphoma, they grow very fast and chemotherapy really doesn't work. So when we have relapsed double hit, triple hit lymphomas, we try to move on with CAR-T immediately and bispecific T-cell engagers. And there is data, immunotherapies do not discriminate whether it's double hit, not double hit, they kill everything. The only challenge is that's where timing is of essence, because they grow so fast that you may run out of time before you got your cart ready. So it's a particular challenge, but what we usually do, we start the process of obtaining the cart, and we give what we call bridging therapy to keep the disease under control. And sometimes bridging therapy, whether it's chemotherapy, drug antibody conjugates, some small molecules, it could be very efficacious and bringing patient into remission or at least low tumor burden, and then cards come in. So that's what pretty much everyone's approach.
Thank you. And we do see a lot of interest in these questions in CAR-T and bispecific T-cell engagers. I know you talked a lot about them, and I think you answered a lot of these questions, but one person is wondering if we know that CAR-T has these side effects. Why would you prefer it as a first option over BiTE, even though you say that BiTE does work well?
Because it's an excellent question. If we're talking 3 years down the line, I may say go for BiTE first. The reason for its data, we're all data driven. So clinical trial for CAR-T, as you can see, the first one was approved 2017. The clinical data is mature. And I know that I can cure patient with CAR-T. And the toxicity is manageable. And really, that's why patient is in the hospital and then they leave the hospital and live their normal life. And I know that if patient is in remission by 3 months, actually most likely cured. Bispecifics, I just do not know. And also with CART, I know very quickly, and I didn't talk much about this time constraint, but we started utilizing not just PET scans, we're also utilizing what we call MRD by molecular means. So you do a blood test and you see if you pick up bits and pieces of DNA in the blood, and the data is accumulating that if it's negative, then the patient is cured. So I may realize it 30 days after CART. So kind of the answer very quickly. Bispecific, it may not be, and we just don't have enough data. And we participated in pivotal trials. So I do have those patients who would have been dead otherwise, and they're probably cured because it's already over a year. I just do not know. We need data. I can't recommend something when we don't have data yet, but absolutely excellent question.
Thank you so much. And I'm going to sneak in one more question on BiTE here. Are there driving restrictions with bispecific T-cell engagers like there are with CAR-T?
Usually it was 28 days.
Mm-hmm.
The rationale, because of ICANS, ICANS happen 5 to 5 to 14 days, most of them, if it happens after infusion. That's the time when patient is still in a hospital. But there were a couple of reports of confusion a couple of weeks later, usually up to, so the safe recommendation is for 28 days. Whether people really follow that or not, I'm not so sure, particularly patients who never had any ICANS. So, but yes.
Well, thank you so much again, Dr. Feldman, for your presentation today and for answering our questions. I want to just remind the audience, I know there are questions left over. If you have any additional questions, please do reach out to our helpline at 800-500-9976. Also, you can reach out via email at helpline@lymphoma.org. Again, you can find all that information at lymphoma.org. But with that, thank you all again for joining. Thank you, Dr. Feldman. Have a wonderful rest of your day.
Thank you.
Take care.
Bye-bye.